Hi Year 11s and welcome to this Digi Explanation. Today we're looking at genetic testing and the developments in biotechnology in this area. Now, let's look at our learning goals for today. These are to be able to 1. Define genetic testing, 2. Outline the 3 main types of genetic testing, and 3. To discuss how genetic testing is developing. Now let's start with number 1. What is genetic testing? Genetic testing includes a range of medical tests that identify changes or abnormalities in chromosomes, genes, or proteins. Now, to give us some more insight into this area, I've interviewed clinical Professor David Ravine, who's a medical geneticist, paediatrician, and pathologist, and works with the University of Western Australia.
Hello, I'm David Ravine. I'm a medical geneticist, and I work with families who have concerns about hereditary diseases, diseases that have been passed down through families, and I'm very much involved with testing.
Professor Ravine's research is centred on Rett syndrome, but in practice he works with a wide range of genetic tests and disorders, which he has seen undergo rapid development in recent years. So to address our second learning goal, let's have a look at the 3 main types of genetic testing that have emerged throughout this rapid development. 1. Chromosomal genetic testing. This is used to analyse whole chromosomes for large genetic changes, such as an extra copy of a chromosome. 2. Molecular genetic testing. This is used to analyse single genes or short lengths of DNA for variations or mutations. 3. Biochemical genetic testing, which is used to study the amount or activity level of proteins as an indication of mutation or genetic variation. Now, to address learning goal number 3, let's go back to Professor Ravine to gain some insight into how genetic testing has been developing.
Since I've been doing genetics, it's grown hugely, and the range of tests has become enormous, and it covers all ages through to late adult life. So it's changing rapidly now. And this change is all driven by research and increased understanding of human genes, human genome. When I first started doing this, genes were theoretical. It was a gene's theoretical concept. It's like an inheritance particle. But now that we know the human genome and the human gene sequence of all genes, the fine detail of all these genes is now being unravelled, and it's a huge change. The pace of discovery in this field is huge, very rapid and accelerating, and it's driven by technology. So only a few years ago we were sequencing genes one at a time, but now we can sequence hundreds of genes at a time, and if you've got the right bit of equipment, you can sequence the whole genome, so 30,000 plus genes at a time. And so the technology, in terms of wet lab technology and also computer science, that fusion has driven this. We have extraordinary amounts of data, which before computers we couldn't possibly handle, but now we can. So that's really what's driving it, and that's exciting, but it also means that a lot of stuff will happen without you find you're suddenly able to do something, and companies want to sell you some bit of kit that allows you to do some even bigger and better genetic test. Now, is it actually a benefit to the person is another question. So genetics, it's kind of, we've moved into new territory, but it's still fraught with challenges brought about by the fact we don't know enough yet.
You should now be able to define genetic testing, outline the 3 main types of genetic testing, and briefly discuss how genetic testing is developing. After learning about genetic testing, I'd now like to challenge you to investigate one of 2 new types of genetic testing. Your options are direct-to-consumer genetic tests or new prenatal genetic blood tests. You'll find more information on Connect. Thank you and good luck.